Hello everyone, my name is Mritunjay Singh and I am a student of B.Tech CSE final year. Today I shall be presenting the topic glycolysis for my presentation under the coursework Biology BBT 7017. So the topic is glycolysis. So what is glycolysis? It is a metabolic pathway that converts glucose into pyruvate. So the free energy which is released in this reaction or in this process, it is used to form the high energy molecules, that is ATP, and the reduced NAD. Glycolysis is a sequence of 10 reactions which is catalyzed by various enzymes. So let us look at the reaction in detail. So glycolysis is a universal catalytic pathway in all living cells. It is also known as EMP. It is a major pathway of carbohydrate metabolism, and all the enzymes involved are present in the cytosol. And the pathway starts with the phosphorylation of glucose to glucose 6-phosphate, which is catalyzed by enzyme hexokinase. ATP is a phosphate donor. Its gamma phosphoryl group is transferred to the glucose. This reaction is irreversible and enzyme hexokinase is inhibited allosterically. It means when the product binds to the enzyme at a site different from the active site and alters its catalytic activity. So this glucose 6-phosphate is then converted into fructose 6-phosphate by enzyme phosphoglucose isomerase, which catalyzes an aldose ketose isomerization reaction. Fructose 6-phosphate then undergoes another phosphorylation by the enzyme phosphofructokinase, forming fructose 1,6-bisphosphate. Similar to hexokinase, phosphofructokinase, also known as PFK, it also catalyzes the irreversible reaction and undergoes allosteric regulation. Fructose 1,6-bisphosphate is cleaved by the enzyme aldolase into two triose phosphates, that is glyceraldehyde 3-phosphate and dihydroxyacetone phosphate. The two trioses synthesized are interconverted by the enzyme triose phosphate isomerase. Oxidations of glyceraldehyde 3-phosphate to 1,3-bisphosphate glycerate occur by the enzyme glyceraldehyde 3-phosphate. The enzyme is glyceraldehyde 3-phosphate dehydrogenase, which is a NAD dependent enzyme. In the next reaction, phosphate is transferred from 1,3-bisphosphate glycerate to ATP, forming ATP and 3-phosphoglycerate by the enzyme phosphoglycerate kinase. This phosphorylation of ADP to form ATP is called substrate level phosphorylation. Since two molecules of triose phosphates are synthesized per molecule of glucose, two ATP molecules are produced at this stage per molecule of glucose undergoing glycolysis. Next step of glycolysis involves isomerization of the compound 3-phosphoglycerate into 2-phosphoglycerate by enzyme phosphoglycerate mutase. The subsequent step involves a dehydration reaction converting 2-phosphoglycerate into phosphoenol pyruvate by the enzyme enolase, which requires either magnesium or manganese for its activity. The phosphoenol pyruvate by enzyme pyruvate kinase is converted into pyruvate, during which phosphate is transferred to ADP to form ATP, which is a substrate level phosphorylation. Out of the 10 glycolytic reactions, 3 reactions are exergonic and therefore irreversible. These reactions are catalyzed by regulatory enzymes, namely hexokinase and PFK, and therefore they are major sites of regulations for glycolysis. So next comes about the energy yield by glycolysis. So the net energy yield by glycolysis is given by glucose plus 2 ATP plus 2 NAD plus 4 ADP plus 4 atoms of inorganic phosphorus give rise to 2 pyruvate plus 2 ADP plus 2 NADH plus 2 hydrogen ions plus 4 ATP plus 2 H2O. Thus, 4 molecules of ATP are generated in the conversion of glucose into 2 molecules of pyruvate. The net ATP production is 2, as 2 ATP molecules are utilized during the process. So this was about glycolysis. Thank you very much.